Welcome to KU Padasala. I am Dr. Preetha. I am working as an assistant professor at University College Tirunelveli. Today I am discussing about in vitro morphogenesis, and it is about the plant regeneration happening during plant tissue culture. Generally, the in vitro morphogenesis in plant tissue culture is of two kinds. They are organogenesis and somatic embryogenesis. Organogenesis is the formation of different types of organs like either shoot or root, and similarly there is the formation of embryo-like structures from the explant tissue during tissue culture, and this is known as somatic embryogenesis. Both these two methods of regeneration, they are again subdivided into two categories. They are direct organogenesis and indirect organogenesis. Similarly, direct somatic embryogenesis and indirect somatic embryogenesis. First of all, let us discuss about the organogenesis. As already mentioned, organogenesis is the formation of organs which is either a shoot or a root, and it is the formation of adventitious organs, and these adventitious organs may be of different kinds like any shoot formation or root formation, and the organ formation directly occurs from the plant tissue, and this plant tissue which is selected for the culturing, it is called as explant. The first history of organogenesis was happened in 1939, and it was from the discovery of White, and he first of all observed the formation of shoot organs in tobacco. Similarly, in the same year, the scientist named Northcote, he also observed the organogenesis from carrot callus, and he observed the root organogenesis. And later in 1957, Skoog and Miller, they have discovered the hormonal control of differentiation in plant tissue culture, and it was a milestone that led to the different types of organogenesis in plant tissue culture. And this is about the hormonal control of differentiation, and it is generally accepted that two different kinds of hormones are regulating the regeneration in plant tissue culture. They are auxins and cytokinins. Auxins are generally stimulating the root formation. Similarly, the cytokinins, they are stimulating the formation of shoots. The ratio of these two hormones are determining a particular kind of regeneration in plant tissue culture. A higher concentration of auxin and a lower concentration of cytokinin is forming the roots, and this root formation is referred to as rhizogenesis. And the higher concentration of cytokinins and lower concentration of auxins are leading to shoot development, and this is known as callogenesis. When the auxins and cytokinins are seen in equal concentration, it is leading to callus development, and it is called as callogenesis. This is the schematic representation of organogenesis, and here the explant, it is giving rise to a callus, and from the callus shoots are produced, and in the shoots the root induction is achieved, and they are giving rise to a plantlet. And the same pathway can be altered in such a manner that the explant can give rise to a shoot directly without a callus phase, and from the shoot root induction and finally plantlet production are achieved. Thus, organogenesis is of two types: direct organogenesis, which is not involving a callus phase, and indirect organogenesis, where a callus intermediate is seen. These are the pictures showing direct organogenesis. Here we can see the direct emergence of shoots from the explant tissue as well as root formation from the explant. And this is the different developmental stages observed during shoot organogenesis from a plant known as Acmella ciliata, and it is commonly known as toothache plant. The second type of organogenesis is indirect organogenesis. Here the explant is giving rise to a callus initially, and from the callus the meristemoids are originating, and these meristemoids are later developing into primordium, like either shoot primordium or leaf root primordium. Thus, the indirect organogenesis is of three phases: de-differentiation phase, which is giving rise to callus. There is the induction of different types of organs, and finally the organ maturation, that is differentiation. And in the indirect organogenesis, a callus formation is essential step, and the culture of callus is called as callus culture. Callus is defined as an unorganized mass of tissue which is produced in response to injury or by means of chemical or physical means. And most of the cells are differentiated in the callus and they are unorganized also. The three criteria for callus culture are aseptic preparation of the plant material, selection of suitable nutrient medium, incubation of the culture under controlled optimum conditions. Here we can see the callus organogenesis from carrot. Here the explants are carrot tissue. They were selected, they were washed properly and subjected to surface sterilization, and after surface sterilization they are cut into suitable sizes and inoculated into nutrient medium, and which evoke the presence of different types of organogenesis from the cambial tissue. That is, meristematic tissues are the choice explant for raising any type of organogenesis. Any plant part can be used as explant for callus formation. It may be either a leaf, shoot, root or stem pieces or floral parts, immature as well as mature seeds, cotyledons, epicotyles, hypocotyles, young seedlings, etc. The color of the callus, it varies depending upon the pigmentation seen in the callus. The texture also is of two types, either compact type or friable type. In the compact type of callus, the cells are highly closely packed, while in the friable type of callus, the cells are loosely arranged. Here we can see the different types of morphogenic responses in callus regeneration. Sometimes the roots are emerging from the callus, sometimes the shoots are also seen, and the pigmentation can arise due to the accumulation of anthocyanin, and after several days, if they are kept without any subculturing, the death of the callus tissue occur, and here we can see the brown coloration, which is an indication of the death of the callus tissue. There are three stages for a callus culture. First stage is induction stage. Here the explants were subjected to callus induction using appropriate growth regulators. Second stage is the proliferative stage. Here the callus cells are undergoing rapid multiplication. And the third and final stage is the morphogenesis stage, where different types of organs are being produced. These pictures are showing the induction stage, proliferation stage as well as the morphogenesis stage of callus culture. The potential application of callus culture are, they are the apt material for vegetative propagation. Somatic embryos can be induced from the callus culture, and they are the starting material for suspension culture. And another important application of callus culture is that they can be utilized for the production of plant derived secondary metabolites, and the production can be scaled up to higher levels using appropriate elicitation strategies. These pictures shows the callus culture in sorghum genotypes. The pictures are indicating that even within the same species, the different genotypes are exhibiting different morphology depending upon the hormonal concentration present in the medium. These are the callus culture of some commercially important plants, that is Podophyllum hexandrum, Azadirachta indica and Linum album. Another important application of callus culture is that they can be utilized for the production of cell suspension culture. This is one of the example in Acmella ciliata. Here the cell suspension cultures were established using callus culture, and the cells were harvested, and these cells were subjected to phytochemical analysis for the elucidation of the most bioactive compound spilandol, and it was present in elevated levels compared to the natural plant population. Callus cultures as well as cell suspension cultures were utilized for the production of artemisinin from Artemisia annua. Also in tobacco plant also, the cell suspension cultures were raised from the callus culture, and the production is scaled up to industrial level using appropriate bioreactor technology. The second type of regeneration in plant tissue culture is about somatic embryogenesis, and it is the formation of adventitious embryos. Here embryo-like structures are produced from the explant, and these are not true embryos. The alternative terminologies for zygotic embryos, non-zygotic embryos or somatic embryos are adventitious embryos, parthenogenetic embryos and androgenetic embryos. The characteristic feature of somatic embryos are that they are bipolar structures, they are having a closed independent vascular system, and they are not connected with the explant tissue or parent tissue. The differences between somatic embryo and zygotic embryo is that somatic embryo is arising from somatic cells, while the zygotic embryos are arising from reproductive cells. Somatic embryos are bipolar, Zygotic embryos are monopolar structures. There is no vascular connection with the explant in the somatic embryo, but in the case of zygotic embryos, it has vascular connection with the parent tissue. Sexual reproduction is not needed for the formation of somatic embryo, while in the case of zygotic embryo, sexual reproduction is essential. And the possibility of somatic embryo induction is happening even in the plants which are not sexually reproducing. But zygotic embryos are induced only in sexually reproducing species. The production time is too short in the case of somatic embryo, but it takes some more years in the case of somatic embryogenesis in some tree species. There are two routes of somatic embryogenesis as similar to organogenesis. Here also we can see direct somatic embryogenesis, which is the formation of somatic embryos directly from the explant, and indirect somatic embryogenesis, where the zygotic embryos are produced from the callus. This is the picture showing direct somatic embryo formation from the explant. In indirect somatic embryogenesis, the explant is first of all giving rise to a callus, and this callus is later giving rise to the somatic embryo, and this callus is called as the embryogenic callus. Thus, in indirect somatic embryogenesis, there is callus induction phase, embryogenic callus development phase, maturation and germination phase. For the induction of somatic embryos, the presence of some auxins are essential in some occasions. That is known as the induction stage. The second stage is known as development stage. Here the embryos are undergoing different developmental stages similar to the zygotic embryogenesis, like globular stage, heart-shaped stage, torpedo stage, cotyledonary stage. And in the germination stage, in usual cases, the somatic embryos are germinating into the plantlets in basal medium, which is devoid of any plant growth regulators. But in some occasions, the presence of some growth stimulating factors are needed. The somatic embryogenesis is differing from organogenetic route of regeneration, and the chief difference is the presence of closed and independent vascular system, and this is one of the feature and unique feature which is differentiating the somatic embryo from the zygotic embryo. Here in this picture we can see different stages of somatic embryogenesis, and the anatomical studies are revealing that there is a closed independent vascular system which is confirming the mode of regeneration occurred via somatic embryogenesis rather than organogenesis. The significance of somatic embryogenesis is that they are ideal plant propagation system like any other tissue culture methods, and the adventitious bipolar embryos, they are offering that there is no need for separate rooting phase, and the cells are arising from single cells, and hence it is of great significance for the production of clonal plants. And also somatic embryogenesis is another approach for the production of disease-free plants. But in somatic embryogenesis also has some limitations. It is not a routine method for plant propagation, because in indirect somatic embryogenesis pathway there is possibility of production of variations due to the callus induction phase. And due to this kind of variations, they are not commonly used as a plant propagation system. But when in the occasions of choosing for diversity, somatic embryogenesis is a better route of regeneration. And both somatic embryogenesis and zygotic embryogenesis as well as organogenesis can be used as propagation system for different culture methods, and it is most beneficial use of somatic embryogenesis and organogenesis is that the whole plants are produced from a single cell. When compared to the merits, there are some disadvantages with regard to somatic embryogenesis. That is, contamination risk is another approach, and it is very common in plant tissue culture system. There is also a risk of mutation, and the responses of tissue culture conditions varies, and all these are depending upon varied plant species. When analyzing the disadvantages, the advantages are standing high, hence somatic embryogenesis and organogenesis can be used as plant propagation system using plant tissue culture techniques. Thank you.